P1KC is a bacteriophage, meaning that it can infect bacteria. Just like many other phages, P1KC has two paths that it must choose between at the start of its infection: the lysogenic cycle or the lytic cycle. In both cases, the phage must attach itself to the host cell and inject its DNA. In this case, P1KC is going to attach itself to a bacterium known as Escherichia coli. If the phage decides to stick around inside its host for a bit longer, it undergoes the lysogenic cycle. To perform lysogeny, the phage DNA that has been injected into the bacterial cell must first circularize. Once the phage DNA is successfully integrated into the bacterial host genome, it becomes a prophage and can continue to replicate, grow and divide with the bacterial cell for as long as it chooses, or for as long as the bacterial cell stays alive. The other path that P1KC can take is the lytic cycle, which you saw in the last animation. What is interesting is that bacteriophages can be very useful for transferring DNA from one bacterial strain to another. This process is called transduction. You should note there are two types of transduction: generalized and specialized. We will not be exploring specialized transduction in this animation, but will provide you with links to more information at the end of this video. In the practical, you will be performing generalized transduction with P1KC and E. coli. The E. coli strain that we will be using for our transduction is AB2828. E. coli AB2828 is an auxotroph due to a mutation in the gene ARA-E. This mutation inhibits the bacterium's aromatic amino acid biosynthesis pathway. This means that it needs the addition of the amino acids phenylalanine, tyrosine and tryptophan to live, as it can't make them itself. By using the phage P1KC as a vector, there is potential for the recipient bacteria to acquire a functioning ARA-E gene from a donor E. coli strain through generalized transduction. Let's see how this happens. Before we do though, note that we won't be going through the whole lytic cycle process as you learnt that in the last animation. If you want a recap of this, please click on the link provided on the screen. However, what we will be focusing on now is generalized transduction, which needs the lytic cycle to happen. We will show you why now. You would recall that once the phage attaches itself to the bacterial cell and injects its DNA, the bacterial DNA is broken down into fragments. The phage then hijacks the host cell machinery to undergo transcription, replication, translation and protein synthesis. This allows the phage to reproduce its DNA and the phage structural components to package this DNA into the phage capsid, ready for release. However, in some cases, bacterial DNA can accidentally get packaged into the phage capsid instead of phage DNA. When the phage progeny is released from the cell via lysis, phages are free to infect other bacterial host cells and perform transduction, mediated through recombination. This includes the phage carrying the bacterial DNA to a donor recipient. This means that phage carrying the E. coli AN180 bacterial DNA can infect an E. coli AB2828 cell and replace the mutated ARA-E gene with the functional ARA-E gene. Now that the E. coli AB2828 cell has a functioning ARA-E gene, it is able to synthesize its own aromatic amino acids and survive happily.